Hi, thank you so much for your interest in our poster. My name is Milton Pividori. I'm a postdoc at the University of Pennsylvania Department of Genetics. We know that correlation coefficients are widely used across different disciplines to identify relevant patterns in data. For example, in transcriptomics, you will very likely correlate the expression of pairs of genes to identify shared function or whether they are part of disease-relevant biological processes. There are several correlation coefficients, but the most popular ones are Pearson and Spearman. However, what we can discover is constrained by what we can detect. And these techniques, Pearson and Spearman, are limited to detecting linear and linear-like relationships. In this poster, we introduced the cluster match correlation coefficient, or CCC, an efficient, easy to use, and not only linear correlation coefficient based on machine learning models. CCC computes a similarity measure between any pair of numerical or categorical data. And to do this, CCC derives clustering solutions for all objects or samples using each variable. And then it compares these clustering solutions to derive a similarity value between variables. This similarity value is between 0, which means no relationship, and 1, which means a strong relationship. Let me show you some examples to see how CCC works. In the first figure on the left, you see 8 panels with scatter plots of 2 generic variables, x and y. The first row corresponds to the classic ISMB quartet, which comprises 4 synthetic datasets with very different patterns, but the same statistics. You can see that the mean, the standard deviation, and Pearson's correlation are the same across these 4 datasets. Let's take a look at 2 of them. For example, in ISMB1, you see a noisy linear pattern that is perfectly captured by CCC. Here you can see how CCC clusters data points using each variable separately, and these clusters match perfectly. In ISMB4, the last one, we have data points with exactly the same value for variable x, but different values for variable y. The only exception is the outlier that you see on the top right corner of the plot. We wouldn't classify this variable pair as a relevant pattern, and you can see that CCC is 0 here, but Pearson and Spearman are quite high. In the second row of this figure, you can see how these coefficients work on other synthetic examples that we generated and were previously observed in transcriptomics. These examples show how CCC can capture nonlinear patterns while being robust to outliers, but maybe the most important part is that these examples highlight how undesirable patterns like outliers or desirable ones, like a biologically relevant nonlinear relationship, can be masked if we use summary statistics alone. We also compared CCC with the maximal information coefficient, or MIC, another nonlinear correlation coefficient. One of the main problems with MIC is its computational complexity. We compared all coefficients regarding this aspect, and you can see the results in the figure below. We found that CCC was up to 2 orders of magnitude faster than MIC. And another property of CCC is that its computation on a single variable pair can be easily parallelized. And in these results, we used 3 CPU cores. We also compared all these coefficients in gene expression data, which you can see on the right of the poster, the right part of the poster. For that, we used gene expression data in GTEx to compute a correlation matrix on whole blood. Then we took for each coefficient 2 groups of gene pairs. First, the highly ranked ones, which we defined as the top 30% of gene pairs with the largest correlation coefficient. And second, a set of gene pairs considered to be not related, and those are defined as the 30% of gene pairs with the lowest correlation values. So we have 6 different categories of gene pairs that can overlap. Then we perform an upset analysis to see how these categories were overlapping. You can take a look at this plot and see the results. There are many patterns that were completely missed by linear-only coefficients. For example, this is one example. CCC captured a strong relationship between genes UTY and KDM6A that you can see in the figure. This relationship is explained by sex differences in gene expression. UTI is in chromosome Y, whereas KDM6A is in chromosome X. And samples with a linear pattern are males, whereas those with no expression for UTY are females. We found this same pattern across all GTEx tissues, not only in whole blood. In addition, we systematically assessed whether gene pairs replicated in 144 tissue-specific gene networks from GIANT, which has no GTEx samples. You can see from the results that gene pairs prioritized by CCC in whole blood were predicted to be specifically expressed in blood-relevant cell types, and also their interaction probabilities were significantly higher. In summary, we show that CCC does not miss widespread, strong linear relationships, while also capturing nonlinear and biologically meaningful patterns that were completely missed by linear-only coefficients. While linear and rank-based only correlation coefficients are exceptionally fast to calculate, not all relevant patterns in biological datasets are linear. We invite you to check out our manuscript and GitHub repo using the QR codes in the poster. There you'll find instructions to install and use CCC. Thank you so much for listening.